Hello everyone, so let's begin with today's class. Um in today's class, we'll study about all the biochemical reactions which are involved in the metabolism in cell and how enzymes control these metabolic reactions. So metabolism in general is divided into comprises of all the reaction which occurs in the cell, and it is largely divided into so metabolism comprises of all the reactions which occur in the cell, and it is mainly comprised of catabolic reaction and anabolic reaction. So we have catabolism and we have anabolism. So in catabolism, we have breakdown. We have breakdown reactions, whereas in anabolism we have synthesis reaction. So in the breakdown reactions, major macromolecules which can be carbohydrates, proteins, or lipids. These are the major macromolecules in the cell, and these are broken down into smaller subunits or simpler molecules. That is, glycogen or starch can be broken down to glucose, basic unit of smaller units like glucose or fructose. Whereas protein is also broken down into amino acids. And lipids are broken down into fatty acids. So the breakdown reaction, which comprises of resolving the higher molecular weight macromolecules into smaller subunits, is known as catabolic reaction. They are part of catabolic reactions, and while breaking down these releases large amount of energy in form of ATP or reducing power. So this energy can be in the form of either ATP, which is the natural currency of the cell, or it could be a reducing power, um, NADH plus. So this reducing power can further be converted into ATP by F0 F1 ATPase in the mitochondria. So in the mitochondria, this reducing power is further converted to ATP molecules, and ATP is used up again in for different chemical reactions and functioning of the cell. So this is general theme of catabolic reaction, whereas in anabolism we have synthesis of reaction. Synthesis reaction like from smaller subunit like glucose and amino acids and fatty acids, we have generation of these macromolecules back again. So these molecules are generated in order to for storage purposes, like glycogen is generated from glucose so that we have a storage molecule. Similarly, fatty acids are converted into lipids, and extra sugar Glucose can also be converted into lipid molecules. And these all conversions, interconversions from within macromolecules and different subunits of glucose and different subunits of each macromolecule like glucose, fructose, amino acids, they are all catalyzed by enzymatic reactions. So all these reaction, all the metabolic reactions are catalyzed by enzymes. All the metabolic reactions are catalyzed by enzymes, and these enzymes play a crucial role in maintaining the proper functioning in the human in the cell. So let's study about carbohydrate metabolism. So carbohydrate metabolism again comprises of, so we have first glycolysis, where glycolysis, as the name suggests, glucose is broken down. So glyco is glucose. Glucose is broken down to, okay, glucose is broken down to give us energy. So this is the glycolysis pathway. After glycolysis, whatever byproduct we get, that is, we get pyruvate. At the end of the glycolysis, we get pyruvate. This pyruvate will further get further converted to acetyl CoA, and this acetyl CoA will go into Krebs cycle, wherein we get ATP production and reducing power, which I was talking about. So even in glycolysis we get ATP. So this is the general theme of carbohydrate metabolism, that first step is glycolysis, second step is pyruvate getting converted to acetyl CoA, and the third step is the Krebs cycle, where acetyl CoA goes into the Krebs cycle. So let's first dive into glycolysis. What is glycolysis? As you can see, we have a glucose molecule here, and glucose, this is the first step of glycolysis and very important. Glucose is converted into glucose 6-phosphate. So this is the first step when the glucose is taken up inside. So we have a cell. So this is the cell, and glycolysis takes place in the cytoplasm. This is very important, that glycolysis takes in the cytoplasm of the cell. So we have glucose molecules in the surrounding, and these glucose molecules are taken up inside in form of glucose 6-phosphate. So glucose is first phosphorylated, and we have a membrane transporters wherein this glucose 6-phosphate is transferred inside the cytoplasm. So the glucose molecule goes inside the cytoplasm. Once this commitment step occurs, where glucose is converted to glucose 6-phosphate, then the glycolysis will begin. So this the first step is very crucial, and it is and we have this enzyme hexokinase. Okay, this is a kinase enzyme, that is, it is transferring the phosphate group. So one ATP is utilized here. There is a utilization of ATP, wherein the phosphate group is transferred onto the glucose molecule. Okay, so this as you can see here, we have a glucose molecule, and we have glucose 6-phosphate, that is, phosphate is transferred onto the 6th carbon here. So the phosphate is transferred onto the C6 carbon, that is by glucose 6-phosphate. So this is the important commitment step for glycolysis pathway. Then glucose is converted into fructose 6 pathway. So and this enzyme is just is a isomerase enzyme, that is, glucose is converted into fructose. So the enzyme is phosphohexose, phosphofructose isomerase. So this is the second important enzyme, that glucose 6-phosphate is converted to fructose 6-phosphate. Also, whenever there is fructose outside the cell, so fructose can also be taken up and can be directly transferred via this step. So fructose can also get phosphorylated, and then the glycolysis resumes from this second step, that is fructose 6 phosphate step. So this is the isomerase enzyme. It is converting glucose to fructose enzyme. Fructose is a 5-membered sugar, as you can see here, and glucose is a 6-membered sugar. Glucose is also fructose is also a 6-membered sugar, but fructose is a ketose sugar, whereas glucose is a aldose sugar. Okay, so we have a ketose sugar here, and aldehyde group is converted into ketose. That's why the name phosphofructoisomerase. And then the next step is where we have one more kinase enzyme involved, that is phosphofructokinase 1. So this is also very important enzyme where the regulation of glycolysis occur. We'll study about regulation in the next slide, how the regulation occurs. So in this step there is one more phosphorylation step, and again one more molecule of ATP is involved. So 2 molecule of ATP is involved in the first part of the reaction. That's why this phase is also known as preparative phase. That is, we are preparing the sugar molecules, either glucose or fructose. We are preparing them into glycolysis. That is, we are basically decorating them with phosphate groups onto, so that they will undergo glycolysis reaction, all these reactions. So we have fructose 1,6 phosphate, which is PFK1. This is also one of the most important enzyme, and it is allosterically regulated, PFK1. Then we have then next step is a cleavage step, where this 6-membered sugar is broken down. Okay, it is broken down into a 3-membered 2 3-membered sugars. Okay, it is broken down into 3-membered not sugars but carbohydrate molecules. So we have glyceraldehyde 3-phosphate and dihydroxy acetone phosphate, and the enzyme which converts this is aldolase. So this is the fourth reaction in which there is a cleavage occurring of this fructose ring. As you can see, this is how the cleavage occur, and this blue part is glyceraldehyde 3-phosphate, and we are remaining with dihydroxy acetone phosphate. Now the glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate and glyceraldehyde 3-phosphate can be interconverted to dihydroxy acetone phosphate, that is DHAP. So we they are both interconverted easily by they can be both interconverted by enzyme. So so now this 2 molecules of glyceraldehyde 2-phosphate, now this is a payoff phase. Why this is a payoff phase? We'll see why this is known as payoff phase. Then the 2 molecules of glyceraldehyde here. So from glucose we get 2 molecules of glyceraldehyde, and it is converted into 1,3-bisphosphoglycerate. Okay, this 1,3-bisphosphoglycerate conversion occurs through this 6th enzyme, that is glyceraldehyde 3-phosphate dehydrogenase. So now we have a dehydrogenase enzyme here. Why? Because we have a conversion of NAD plus to NADH. So we are getting a reducing power. So that is why this is a payoff phase. That is that means that we'll get reducing power, or we'll get ATP. So whatever preparative phase or whatever ATP were consumed here, they will be paid off in the later phase of glycolysis. So glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, 2 molecules of 1,3-bisphosphoglycerate. So we'll get 2 molecules of NADH and H plus. Then the next step is 1,3-phosphoglycerate. It's converted into 3-phosphoglycerate. Okay, so again we get 2 molecules of ATP. So whatever 2 molecules of ATP we used before, we got those 2 molecules of ATP back. So and this enzyme which converts is phosphoglycerate kinase. Then from 3-phosphoglycerate we have 2-phosphoglycerate, again by there is by phosphoglycerate mutase. So the phosphate group from third position is now shifted to second position. So this is a mutase enzyme which converts this phosphoglycerate mutase to 2-phosphoglycerate then we have a hydrolysis reaction which is carried out by enolase. Okay, this hydrolysis reaction, what happens here, we'll get PEP. Okay, so phosphoenol pyruvate. So as I mentioned before, the final step is the pyruvate, so we are getting there. So and the final step is pyruvate kinase. So again there is a kinase enzyme involved, and we'll get production of ATP. We'll get production of ATP here. Okay, so finally overall reaction of glycolysis could be that we have a glucose molecule. Okay, and this glucose molecule is converted to pyruvate. Okay, this glucose molecule is converted to pyruvate. Basically we'll have 2 molecules of pyruvate, right? These are all 2 molecules here, so we'll get 2 molecules of pyruvate. And apart from glucose, what do we have? What are we getting? We are getting 2 molecules of NAD plus. NAD plus is involved, so we are getting 2 molecules of NADH plus H plus, that is 2 reducing power, 2 hydrogen reducing power, and 2 molecules of ADP. Okay, we'll get 2 molecules of ATP. So I'm writing only 2 molecules of ATP because we have used 2 molecules here, and that is why only 2 molecules of so these ATP molecules will get cancel off it, so only 2 molecules of ATP is formed. Okay, but it's all multiplied by 2, so yeah. So from reducing power we generally get, so 2 NADH H plus. This reducing power gives us approximately how much? It will give us 3 ATP molecules. Okay, it will give us 3 ATP molecules. So the overall ATP molecule from yes, so the overall ATP molecules which we get are 3 ATP and 2 ATP, that is total total 5 ATPs. Okay, total 5 ATPs. Sorry, so 3 ATP, so this will be total 6 ATPs because we have 2 NADP. So 1 NADH will give us 3 ATPs and 2 NADH will give us 6 ATPs. So this will be 6 plus 2, that is 8 ATP molecules. So from glycolysis we have 8 ATP molecules. Okay. So this is the glycolysis balance sheet I was talking about. So in the preparative phase, we had minus 2 ATPs here involved. Okay, minus 2 ATPs. These 2 ATPs were used. Okay, they were not created, but 2 ATPs were used. Then in step 6, we have 2 ATPs each. So we get 2 NADH plus, and from each NADH will give us 3 ATPs. So total ATPs will get 6, and we have two ATP generation steps. So the total ATP will be generated, balance ATPs will be 8. Okay, after correcting these two used ATPs. Now, how the glycolysis is regulated? So glycolysis is regulated by a hormone known as insulin. So whenever the insulin, so the first step is the insulin binds to the receptor. So whenever there is a sugar outside the cell, the insulin will come and it will bind to the insulin receptor, as you can see here. So this is the first step, that is insulin will come and insulin will bind to the receptor. After binding to the receptor will get activated, okay, and it will promote the recruitment of glucose. That is, this is the second step, that recruitment of glucose transporters. So it will promote this glucose transporters. So they will have so these glucose transporters basically will phosphorylate glucose, and it will take up the glucose molecule from the inside. So the second step is promoting this glucose transporters. These are the glucose transporters. They will be promoted and they will be exported into the plasma membrane. The third step is that now glucose will finally enter the cell as glucose 6-phosphate. So glucose will enter the cell as glucose 6-phosphate through the transporters. And once it has entered into the glucose, now what will happen? Inside glucose, once the levels of glucose outside are low, okay, all the glucose has now taken up inside. Now the level of glucose is low outside extracellularly. So the insulin level will decrease, and again the transporters which were now expressing these promoters, they will come back. Okay, they will come back intracellularly and they will be recycled. They will be either degraded or they can be recycled. So this is they can be fused to an endosome, or they could be phagocytosed. Okay, so these can be just degraded. They can either be recycled or just degraded into the endosome. So this is how the glycolysis is regulated via hormone. And enzymatically, how do glycolysis regulated? So as I mentioned earlier, the enzyme, there are two enzymes. Okay, the two enzymes are phosphofructokinase 1 enzyme, and the other is this pyruvate kinase enzyme. So what happens? So we have so when the blood glucose levels are very low, okay, the blood glucose levels are very low. So what will happen at that time? So we have conversion. So pyruvate kinase enzyme converts phosphoenol pyruvate. So this is the last step of the glycolysis. So it will convert phosphoenol pyruvate to pyruvate, okay, and this reaction is positively regulated by fructose 1,6 bisphosphatase. Okay, so fructose 1,6 bisphosphatase, what it will do, it will remove the phosphate group. Okay, it will remove the phosphate group, and it will it will make it either active or inactive. Okay, so this is how the low blood glucose levels are regulated with respect to pyruvate kinase enzyme. And in case of phosphofructokinase, so in case of phosphofructokinase, this enzymatic regulation, so we have this fructose 1,6 bisphosphatase enzyme. So this enzyme is very important. So this phosphofructokinase 1, what does it do? It converts fructose 6 phosphate to fructose 1,6 phosphate. Okay, and this bisphosphatase, what it will do, it will remove this phosphate group. Okay, it will remove the phosphate group, right? This phosphofructokinase, what it will do, it will remove. So this is the reaction. So we have two enzymes. So we have a kinase enzyme, which is a part of glycolysis, and it is adding the phosphate group. It is the kinase enzyme adds or transfers the phosphate groups. Okay, and we have a phosphatase enzyme, and this phosphatase enzyme, what it will do, it will remove the phosphate group. Okay, it will remove the phosphate group and it will maintain this phospho 6 phosphate level. So this enzyme, as you can see, it is removing the phosphate group. Okay, it is removing the phosphate group, and thus it is converting phosphofructokinase level 1. So yeah, so it is so this is how it is converting so this is allosterically inhibiting PFK. Okay, it is allosterically inhibiting. So phosphorylation is causing either to phosphorylation of phosphofructokinase is basically, so we have phosphorylation of PFK. Okay, so this phosphorylation of PFK, whenever there is a phosphorylation of PFK, what will happen? We'll have conversion of fructose 6-phosphate to fructose 1,6-bisphosphate, and whenever this phosphorylation is removed, we have the reverse reaction. Okay, so whenever there is phosphorylation, we'll have a forward reaction, and whenever this phosphorylation is removed, we'll have a backward reaction of, so this phospho 1,6-phosphate is converted into fructose 6-phosphate. So this is how allosterically this enzyme is getting regulated. Now we move on to Krebs cycles. Once we have pyruvate generated, so this pyruvate will now go into this pyruvate in the cytosol only. It will get this pyruvate will be generated in the cytosol. Okay, and it is converted into acetyl CoA by a complex which is known as pyruvate dehydrogenase complex. Okay, it's why it is a complex. It's not a single enzyme, but it's a mixture of 3 different enzymes, and these 3 different enzymes are converting this pyruvate into acetyl CoA. And so this is a coenzyme A. Okay, so this is very important step, this pyruvate dehydrogenase complex step. That is, this step is committing the acetyl CoA. It's committing that there will be Krebs cycle. Okay, it's committing the pyruvate to go into Krebs cycle. That is, after conversion of pyruvate to acetyl CoA, the acetyl CoA now has to go into the Krebs cycle. So this is the commitment step or the regulation step of the Krebs cycle. So once it goes into acetyl CoA, so it's a cycle. That is, at the end of the cycle, we'll get our acetyl CoA back. Okay, so what is happening? Acetyl CoA plus oxaloacetic, we get citrate. Okay, we get citrate, and then we have citrate to cis aconitate by aconitase enzyme. Then we have cis aconitase to isocitrate by again aconitase enzyme. So these are just mutase or isomerase kind of enzyme. Then we have, from isocitrate, we have oxalosuccinate. So this enzyme is isocitrate dehydrogenase. So whenever there is dehydrogenase, we'll have a reducing power generation. So this is our first step of getting reducing power in Krebs cycle. Okay, now this reducing power will also is a energy currency, as we know, because one NADH molecule will give us 3 ATPs. Okay, so this is kind of a energy currency for us. Then oxalosuccinate is converted to alpha ketoglutarate. Okay, and we have a loss of CO2. Okay, so this loss of CO2 will be taken care of by other reactions. Okay, we'll study about urea cycle, that how this CO2 is taken care, because CO2 is very harmful to the cell, so this has to be taken care of. So this will be taken care in urea cycle. We'll study this at the end of the class. Then we have conversion of alpha ketoglutarate to succinyl CoA, and again the enzyme is alpha ketoglutarate dehydrogenase. So as we know, dehydrogenase, so we'll have another molecule of NADH. So we have two molecules of NADH getting formed. Okay, two molecules of NADH getting formed, and we'll get how many? We'll get 6 ATP. Okay, 3 plus, we'll get 6 ATPs as of now from the Krebs cycle. And then from succinyl CoA to succinate, we have a GTP, and GTP in the energy currency is equivalent to ATP. Okay, it can be interconverted, and they are equivalent as ATP only. Okay, so then we have from succinate, we have fumarate by succinate dehydrogenase. So the enzyme name is named after reactant and dehydrogenase if there is a reducing power. So here also isocitrate dehydrogenase, we have a substrate and dehydrogenase enzyme. Similarly here alpha ketoglutarate, which is a substrate, and dehydrogenase. So again we have FADH2. So this FADH2 reducing power is equivalent to 2 ATP molecules. Okay, it has slightly low energy currency value in forms of ATP. Okay, so it gives only 2 ATP molecules when these protons go into mitochondria for ATP generation through F1 F0 ATPase. Then finally we get oxaloacetate back, and this oxaloacetate can further react with acetyl CoA molecules which are generated from pyruvate. So as we know from glucose, from one molecule of glucose, okay, one glucose molecule, we'll get two pyruvates, okay, and so let's not forget this reducing power also. So we have two NADH here getting generated from one glucose molecule. So one glucose molecules will get here we'll get 2 NADH, okay, which will give us 3 ATPs, and then here this all will get multiplied by 2, okay, because this cycle will go twice, right, because there are two pyruvate molecules, so we'll get 2 acetyl CoA molecule, and this 2 acetyl CoA will interact with 2 oxaloacetate molecules, so we have to multiply everything with 2. So again GTP will also be 2. So let's see what is the final balance sheet of Krebs cycle. So okay, so this is the balance sheet of Krebs cycle. So we have how many? So from Krebs cycle, as you can see, production of acetyl CoA. So number of ATP produced. So this is acetyl CoA. It was given us NADH. So pyruvate to acetyl CoA had given us 1 NADH. So it will give us 3 ATP. Okay. And in alpha ketoglutarate also we got it from NADH only, reducing power, so again 3 ATP. And from succinyl CoA again NADH, right, NADH was involved, so again 3 ATPs, and then we got 1 GTP which is equivalent to ATP only, so 1 ATP here. Then we had FADH2 which is equivalent to 2 ATP molecules, okay, so 2 ATPs, and then finally we get oxaloacetate. Then here also we are getting 3 ATPs from NADH. Okay, so we get total 15 ATP molecules, but we have to multiply this by 2. Why? Because we get 2 pyruvate, then we'll get 2 acetyl CoA. Okay, so we have to multiply this by 2. So in glycolysis 2 molecules of pyruvate are generated, and hence total we have to multiply it by 2. So total 30 molecules of ATP will be generated. So from Krebs cycle we'll get 30 molecules of ATP. Okay, and we got 8 molecules of ATP from glycolysis, so total 38 molecules. Okay, total 38 molecules of ATP will get from glycolysis plus Krebs cycle. Okay, total 38 molecule of ATP will get from glycolysis and Krebs cycle. So if we so this 38 molecule will only occur in case of aerobic respiration. Okay, this 30 molecule, whatever reducing power which um, uh, reducing power getting converted to ATP, that only occurs in aerobic respiration through electron transport chain, right? Electron transport chain, ETC, and this will only occur in case of aerobic. Whereas for anaerobic pathway, we'll have only 2 molecules of ATP, that is by glycolysis. So through anaerobic system we'll have only 2 molecules, whereas from anaerobic we'll have 38 molecules of ATP. Now how the Krebs cycle is regulated. So Krebs cycle can be regulated by so here this is the major regulation step. So the byproducts regulate the enzymes. Okay, so the byproducts of this pyruvate to acetyl CoA is acetyl CoA, and this acetyl CoA further goes into fatty acid oxidation. So fatty acid oxidation is one of the negative regulator, acetyl CoA is also negative regulator. So whenever there is lot of byproduct formation downstream, it will have a feedback control on the enzyme. Okay, and if there is lot of ATP and reducing power already present in the cell, so it will just inhibit, it will tell the cell to stop, that we no longer need this Krebs cycle going on, so we need to have a negative regulation. So these are all part of these are all negatively this negatively regulate the enzyme. Okay, that is pyruvate dehydrogenase complex. And what are the positive regulators? Suppose we have ADP molecules which are very high, NAD molecules and fatty acids. Okay, we have lot of fatty acids and calcium. So calcium is one of the important regulator, as you can see here. So these are the positive regulators. So when ADP molecule will be high, ADP and NAD molecule will be high whenever there is a depletion in the energy currency of the cell. So whenever there is such depletion of the energy currency, we require Krebs cycle to generate the ATP. Okay, we will require Krebs cycle to generate the ATP, and that ATP will be further used. Okay, it will have this balance. So ADP plus PI will give ATP. So whenever the energy currency is low, we'll have a higher amounts of ADP. Low energy will have higher values of ADP, so it will positively regulate. So we want the Krebs cycle to occur in order to get the energy currency. Okay, then it is again, so ATP, NADH for these higher energy currency molecules. So whenever we have higher ATP molecules, so we have enough energy currency, they will negatively regulate this oxaloacetate to citrate. That is the first step of the Krebs cycle. So they will not allow the Krebs cycle to occur. Okay, and all the energy consuming process, that is isocitrate to alpha ketoglutarate. Okay, whenever there is energy conversion process, that in form of reducing power or ATP, those steps are highly regulated. So we have a NADH and ADP controlling this step as well. Also alpha ketoglutarate to succinyl CoA, where NADH, which is a reducing power generation step, is also controlled. Okay, so this is how the energy balance occurs in the Krebs cycle. Now we move on to lipid metabolism. So in the lipid metabolism, whenever we consume this fatty acids, um, fatty diet which is rich in fats, so it could be any oily food or fish or other non-veg. It could lead to generation of triglyceride in the intestine. So this triglycerides are further digested by the in the intestine by the lipases. Okay, and this lipases are released from the bile salts. Okay, so we have a bile salts in the gallbladder. So whenever we have high fatty meal, the gallbladder will emulsify this fatty meal by degrading the triacylglycerols, okay, in the intestine. So we'll have a large lipid molecule converted into small triacylglycerol, as you can see. So these are the or degraded form of huge lipids, okay, large molecular lipids. These are the degraded forms, and these degraded forms are taken up by the intestinal linings. Okay, they are taken up by the mucosal intestinal linings of the ear. So they are taken up in the bloodstream. Okay, they are taken up in the bloodstream from here. Okay, and then they go into, and then they are transferred into. So the next step is they are transferred. This glycerol are packed with cholesterols. Okay, and we have apolipoproteins. So these proteins are a combination of protein and lipids. That's why the apolipoproteins. Okay, and this apolipoproteins will form a kind of micelle, and this micelle again are known as chylomicron, which will transfer into the blood. Okay, this will transfer into the blood, and it will go into the adipocytes. Okay, adipocytes are the storage tissues where we store fats, and these and they are then further transferred, okay, into adipocytes, where it will either be stored as fats. Okay, there will be a fat storage, or there could be a fatty acid oxidation. Okay, there could be a degradation, and this degradation is known as beta oxidation. Okay, this degradation is known as beta oxidation. So we'll study about this catabolic reaction of lipids. So the storage fats are the energy reservoirs which can be further degraded whenever we require energy, or if we are doing a high intensive workout, or we are utilizing large amounts of energy. These storage fats are used as the energy reservoir, and they are broken down whenever we need huge amounts of energy, and they are broken down in by beta oxidation reactions. Okay, so what is this beta oxidation? So we have fatty acyl chain. So this is our one of the fatty acyl chain. Okay, and as you can see, it's a how many membered carbon? We have 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15. So it's a pentatechanoic acid. It's a pentatechanoic, that is 15 carbon fatty acid. And why do we call it beta oxidation? So this is our this is our carbonyl carbon, and carbon attached to the carbonyl carbon is the alpha carbon, and the carbon besides the alpha carbon is beta, gamma, delta, so on. This is how the nomenclature goes on. So this beta oxidation occurs when the bond between alpha, the covalent bond between alpha and beta carbon is broken down. Okay, so the fatty acyl chain is degraded through beta oxidation. That means this bond will be broken down. Okay, so similarly the breakdown will occur after every 2 carbon. Okay, this is how the breakdown will occur. Okay, and finally we'll have a 1 carbon or acyl carbon which is remaining with us. So this is how actually the reaction takes place. So we have a fatty acyl chain which could be of n number of length. Okay, and we have a CoA and we have a acyl CoA. So this is the enzyme, the first enzyme in the beta oxidation pathway. Okay, so this first we'll have a acylation. We have a acyl CoA attached to the fatty acyl chain. Okay, and we'll get a fatty acyl CoA molecule. Okay, now this fatty acyl CoA is a charged. So this is a high. So this tilde here, this tilde refers to a very high energy bond. So whenever this acyl CoA binds to fatty acid, we have very high energy bond. So this high energy bond will prime the beta oxidation step. So it will prime the cleavage between C alpha and beta. Okay, C alpha and C beta. So having a acyl, okay, having a acyl high energy bond here, okay, it will be easier for the cleavage of beta oxidation. So what are the steps in beta oxidation? So this is the stage 1 of beta oxidation, um, wherein what this occurs in the cytosol. Okay, this occurs in the cytosol where the fatty acyl chain are transferred. Okay, and it goes and beta oxidation occurs, and we'll get it is broken down into acetyl CoA molecules. Okay, uh, if we have a 16 carbon fatty acyl chain, okay, it will broken down into 8 acetyl CoA. It will be broken down into 8 acetyl CoA. Acetyl CoA is a 2 carbon molecule. So 16 carbon fatty acyl chain will be broken down into 8 acetyl CoA molecule, and this acetyl CoA will further go into Krebs cycle. So this is how we get so beta oxidation is nothing but the fatty acyl chain is broken down into 2 carbon acetyl CoA molecules. Okay, so this will become one acetyl CoA, this will become another. Okay, like that it will get broken down into different acetyl CoA molecules. Okay, and those acetyl CoA will further go into Krebs cycle. Okay, so that's why acetyl CoA is one of the important regulator of Krebs cycle. And from Krebs cycle we will get reducing power, and also we'll get ATP as well. Okay, we'll get reducing power and ATP. And the stage 3, this occurs in the mitochondria, right? This occurs in the mitochondria where we have the oxidative phosphorylation ETC, where all the reducing power will get converted into the ATP. Okay, now beta oxidation is regulated by this carnitine shuttle. So what is this carnitine shuttle? So in order to have this whenever this acyl chain linked to this higher molecular weight acyl CoA chain of fatty acid, it has to transfer from the cytosol to mitochondria. Okay, so we have outer membrane, inner membrane space, and we have the matrix of mitochondria. Okay, whenever it has to be transferred into the, so we have a carnitine molecule. So this carnitine molecule will first have so this carnitine molecule will get transferred onto this fatty acyl chain. So this carnitine molecule get transferred here, as you can see. So we have a carnitine fatty acyl chain here, and then this will easily get transported from a transporter. Okay, so this acyl carnitine translocase will translocate this inside, and then this carnitine will again get released, and it shuttles different fatty acyl chains in this form. Okay, so this is how it gets transferred into the mitochondrial matrix. Okay, this is how it gets transferred into this mitochondrial matrix to get where the beta oxidation occurs. So these are the steps of beta oxidation, as you can see. So we have so this is the alpha beta carbon, and then acyl CoA dehydrogenase first will form a dehydrogenase step, that is this single bond will get converted into a double bond. Okay, then we have enol CoA hydratase. Okay, it will have a hydroxyl group. Okay, at the beta carbon, so there will be a cleavage here. So we need to have a cleavage here. Okay, so we are converting this enzyme into dehydrogenase. Then Acyl transferase, what it will do, it will cleave this and will have this acetyl CoA molecule, and this fatty acid chain now has -2 carbon. Okay, now this -2 carbon, why? Because we have a acetyl CoA formation from this chain, so this chain has broken down from here and we'll get our first acetyl CoA molecule. Similarly, this step repeats until all the beta cleavage, alpha beta cleavage takes place. Now, how does the fatty acid metabolism is regulated? So whenever we are consuming, whenever there is high blood glucose, we are consuming diet rich food, glucose rich food, what will happen? It will trigger the insulin to take up the, okay, it will take up the glucose, and it will, what it will do, it will also activate, okay, this ACC molecule. Okay, so this ACC will get activated in presence of, so in whenever there is phosphatase, insulin, whenever there is high glucose, it will get the phosphate group will get removed and it will get activated, and it will convert acyl CoA to malonyl CoA. Okay, and will have a fatty acid chain formation. Okay, this will lead to fatty acid synthesis. So high glucose will get converted into fats. Okay, whenever we have low blood glucose, what will happen? The glucagon will get broken down, and we have different enzyme pKa and AMPK. Okay, so they will convert this active ACC into inactive ACC. Okay, and whenever this becomes inactive, there will be a stopping in fatty acid synthesis. Okay, so there won't be any fat storage. Okay, because we are low on blood glucose level, so whatever fats we have, we need to have a fatty acid degradation. So then again, in the mitochondria we have carnitine acyl transferase, that also will have a inhibition here in the mitochondria, where we so in mitochondria we have beta oxidation. So if we are low on blood glucose, the carnitine acyl transferase, it will trigger this fatty acyl CoA transfer, and it will trigger beta oxidation. So we'll have more of energy, or we'll have energy currency. Now next we move on to amino acid metabolism. So amino acids are synthesized, so we don't have we don't have amino acid pathways as such, but they are synthesized in glycolysis step as well as in Krebs cycle step. So as you can see, from glucose we have different amino acid synthesis. So from glucose we have glucose can be converted to ribose 5-phosphate, and from that we have a histidine. Okay, so from glucose we have histidine getting converted. So from glucose we have histidine getting converted, amino acid, and so from 3-phosphoglycerate, that is 3-PGA, from 3-PGA we have serine. We have serine synthesis, and then we have glycine and cysteine. Okay, so these are the amino acid converted from 3-PGA. So these are the amino acid which we get from. This is the glycolysis. Okay, this is the glycolysis pathways. And from phosphoenol pyruvate we have tryptophan, phenylalanine and tyrosine. So from phosphoenol pyruvate, that is PEP, we have all the aromatic amino acids. We have formation of all the aromatic amino acids, and from pyruvate we have formation of large amount of amino acids. That is, from pyruvate we get alanine, we have valine, we have leucine and isoleucine. Okay, and again from the Krebs cycle, so this is our Krebs cycle. So from Krebs cycle also we have oxaloacetate, which also synthesizes. So from this oxaloacetate step, so all these are the precursors for these amino acids. So from glucose we have synthesis of histidine. So glucose is a precursor of histidine. 3-PGA, 3-phosphoglycerate, is a precursor of serine, glycine and cysteine and so on. So from oxaloacetate, which is a precursor for aspartate, okay, and from aspartate we can further get aspargine, okay, aspargine, and we have methionine, threonine and lysine, okay. So these are the amino acids from oxaloacetate. And then we have alpha ketoglutarate. From alpha ketoglutarate also we first get glutamate amino acid, acidic amino acid, and then from here also we have glutamine and proline and arginine, okay. So this is how the amino acids are synthesized. So not all 20 amino acids are synthesized in humans, okay. Not all 22 amino acids are synthesized. Some are need to be taken up in form of diet, okay. So those are the essential amino acids. So those are essential amino acids which are taken up from diets. So we our body is not able to synthesize those amino acids, and thus we need to have the essential amino acid through diet. So this is only the difference between essential and non-essential. So these are the all non-essential amino acids. So we have tyrosine, which is missing, okay. So tyrosine is the essential amino acid, and that has to be taken up from the diet. So these are the amino acid families based upon the precursor, as so histidine we got from glucose, and then we have aromatic amino acid from phosphoenol pyruvate, and then serine is synthesized. So serine is the first amino acid, and from serine we get cysteine, glycine. Similarly, we have aspartate and glutamate amino acid. So glutamate comes from oxaloacetic acid, and aspartate comes from alpha ketoglutarate, and we have this n number of amino acids which are synthesized downstream to this amino acid. Then again the amino acids can be regulated, okay. These are regulated at various steps of synthesis. So the major step is that which converts glutamate to glutamine is glutamine synthesis, okay. So this enzyme glutamine synthesis is regulated by different forms. So it is regulated by glycine, it is regulated by alanine, it is regulated by CTP, okay. It is regulated by histidine, it is regulated by glucosamine 6-phosphate, it is regulated by carbamyl phosphate, okay. So it is regulated by this 8 molecules, okay. This is regulated by 8 molecules, okay. So whenever the downstream concentration of these byproducts, so all these 8 molecules are the byproduct of the glutamine synthase. So whenever the byproduct, there is a downstream byproduct of this glutamine synthase, the concentration exceeds, they have a feedback regulation, okay. This is known as feedback regulation. Feedback regulation of the product itself inhibits the enzyme. So this is how amino acid is regulated. And so the urea cycle is one of the important cycle, because whatever CO2 or ammonia or NH2 plus is getting converted through Krebs cycle or amino acid synthesis, this needs to be taken care of. So we have a urea cycle. So this urea cycle, as you can see, so we have amino acid, okay, and then we have ammonia generated here, Okay, NH4 plus. Ammonia is getting generated through amino acid synthesis, and we have glutaminase enzyme which removes ammonia. Okay, so we have ammonia getting generated from amino acid, and we have CO2 getting generated from Krebs cycle, okay, from mitochondria. So the first step of this urea cycle is ornithine plus carbomyl phosphate. Okay, carbomyl phosphate, it gives us citrulline. Okay, this is the first step, that ornithine plus carbomyl phosphate gives us citrulline, and then this further citrulline gets converted to citrulyl AMP intermediate and arginosuccinate fumarate, and then from fumarate again we have ornithine synthesis. So this is how this ammonia which is released from the amino acid metabolism is getting fixed into is getting converted to urea, and then this urea can be excreted. Okay, this urea can be excreted from the system. So this is the general urea cycle. So now we move on to the assignment questions. The step in glycolysis that results in the production of both NADH and ATP is. So only one step which gives us NADH reducing power as well as ATP, that is the energy harvesting of glyceraldehyde 3-phosphate. Okay, so C is the answer. So from this reaction we have NADH, one molecule of NADH plus ATP generated. Okay, the high energy compound formed directly from dehydration of 2-phosphoglycerate is. So from 2-PGA, 2-phosphoglycerate, we get, so from 2-PGA, 2-phosphoglycerate, we get PEP. Okay, phosphoenol pyruvate. Okay, it's a high energy compound because it has a phospho group. Okay, it has a phospho group. Okay, so this results in formation of this phosphate group formation results in formation of 2-PEP. Okay, so we have phosphoenol pyruvate generation. In order to slow down glycolysis, when cellular energy levels are high, the PFK are inhibited by. So PFK, whenever the cellular energy levels are high, so we'll have higher molecules of ATP, the energy currency will get high, and also along with that citrate will also be high. Why citrate will be high? Because if there are high levels of energy, there will be unused citrate in the cell if there is no Krebs cycle going on. So it will lead to accumulation of citrate. Okay, so ATP and citrate is the answer. The step in Krebs cycle that involves substrate level phosphorylation is. So substrate level phosphorylation occurs at whenever there is a GTP, when there was a GTP formation, and that occurs when the conversion of alpha ketoglutarate to succinyl CoA, we get one molecule of GTP. Okay, the intermediate that connects the lipid metabolism to Krebs cycle is, so the intermediate is citrate. Okay, the citrate is a first step where the acetyl CoA, okay, acetyl CoA along with oxaloacetate forms citrate molecule. So it is the first step, and that connects to that of the, so acetyl CoA connects to that of the lipid molecule. So for the formation of citrate, we have acetyl CoA plus oxaloacetic acid. Okay, and this gets converted to citrate. So this is the first intermediate into lipid metabolism as well as Krebs cycle. So this will either go into lipid metabolism or it can go into Krebs cycle. The intermediate that connects the lipid metabolism, the activity of alpha ketoglutarate dehydrogenase in Krebs cycle is inhibited by. So alpha ketoglutarate dehydrogenase, so as the name suggests, this dehydrogenase enzyme, so the byproduct is alpha ketoglutarate dehydrogenase byproduct is, so alpha ketoglutarate gets converted to succinyl CoA, right? And we have it's a dehydrogenase, so we have a NADH2 also, NADH plus H plus. We have a proton. Okay, we have a proton reducing power generate. So this enzyme can be regulated by both byproducts, by succinyl CoA as well as NADH. Okay, so these are the byproducts generated by this enzyme. So alpha ketoglutarate dehydrogenase converts this into succinyl CoA and NADH plus. So these both the byproducts are responsible for regulating. Whenever there is high levels of succinyl CoA and NADH, it will negatively regulate the activity of alpha ketoglutarate dehydrogenase enzyme. Okay. The enzymes involved in breakdown of triglyceride during lipolysis is, protease and amylase, it will break down protein and carbohydrate. Hormone sensitive lipase and lipoprotein lipase, yes, they will break down lipids. Amylase pectinase, again it will break down carbohydrate, amylose and pectin. Catalase and cellulase, no, so the correct answer is B. The final result of thiolysis reaction in beta oxidation is catalyzed by acyl CoA acyl transferase thiolase is. So the it's the production of acetyl CoA and a shortened fatty acid. So beta oxidation first step is so this acyl transferase, it will chop off 2 carbon acetyl CoA, it will chop off 2 carbon acetyl CoA. So whatever the fatty acid carbon chain it will be CX, it will get -2. It will get converted, it will get shortened by 2 carbon. The insulin influences fatty acid metabolism during high blood glucose levels by it regulates by allosterically by allosterically activating acetyl CoA. Okay, acetyl CoA ACC, as we have seen before, it will allosterically activate this acetyl CoA carboxylase ACC enzyme. Okay, the step in fatty acid oxidation that is affected by the regulation of carnitine shuttle is entry of fatty acid CoA into mitochondria, synthesis of malonyl CoA, activation of ACC, inhibition of PK. So carnitine shuttle takes place in the transport of fatty acid CoA into the mitochondria. So this is whenever there is carnitine shuttle, there won't be any entry of fatty acid CoA into the mitochondria. So if there is no fatty acid in the mitochondria, there won't be any beta oxidation. As simple as that. Primary regulatory mechanism of controlling the urea cycle activity is feedback inhibition, transcriptional regulation, allosteric regulation of enzyme, post-translational modification. So urea cycle activity is allosterically regulated. Okay, so the first step in the urea cycle where involved in with the citrulline, it is allosterically regulated. The primary nitrogenous waste produced as a byproduct of metabolic processes are, so the primary nitrogenous waste. So nitrogenous waste means it will contain nitrogenous compounds. So only nitrogenous compound are ammonia and urea. Glucose, lipids, carbohydrate, they don't have any nitrogenous compound. The correct answer is A, ammonia and urea. The coenzyme that is primarily utilized by aminotransferase during amino acid biosynthesis is aminotransferase is the coenzyme that is involved is pyridoxal 5-phosphate PLP. The enzyme that facilitates the reversible interconversion between alanine and pyruvate. So this enzyme is alanine aminotransferase. Okay, this enzyme will convert from pyruvate to alanine. Okay, reversible interconversion. The pathway that serves as the central pathway for biosynthesis of amino acid is shikimate pathway. So this is it.